Okay, another group that wanted to be featured for first years here in class is this pre-genetic counseling club. So if you have any interest in this field at all, there is a club on campus geared towards this. Lily Lehigh, who some of you might have for a TA in lab, she's also a tutor. She is the head of this club, so if you're interested, send her an email. I think they try to meet, I want to say like once a month, but I could be completely wrong on that. All right, so I wanted to start off class today by clarifying some inconsistencies we've seen with drawing atoms. All right, this is on me. This is like the deep recesses of my mind, and I had totally forgotten about this. So as you might have noticed, we have seen a couple examples where, for example, this potassium atom, this third shell can hold up to 18 electrons, but in this case, it's only holding 8 before going to that fourth shell. Right? And I wanted to explain why this is, though I don't expect you to memorize this. Right? This is more like chemistry class stuff, but I just wanted to throw it out there so you understand why it is you're seeing it in this arrangement. So these electrons, they have subshells, right? Subshells are different than orbitals, right? And they're essentially groupings of electrons within the shells, and it kind of determines how these shells will fill up with electrons, right? Now in the smaller atoms, they fill up exactly like we have been learning and how I would expect you to know for the purpose of this class. But once we get beyond the third electron shell, things start to get a little funny. So I thought we'd walk through how this would fill up for our potassium atom. So we have these different subshells, right? These different subshells, they can contain different numbers of electrons, right? So the S can have 2, the P can have 6, D is 10, and F is 14. So we're going to follow the path that they fill up using this diagram for potassium. So first of all, we have our first electron shell, right? That means we're going to start with this 1s, the S subshell. You know it can hold 2 electrons. We're going to put 2 electrons in there. Great. We have filled up that first electron shell. All right, the next one that we want to fill would be the 2s, right? There it goes. The 2s subshell, right, in that second electron shell. So I'm going to put 2 electrons there. Right? It's telling me next that I'm going to have that 2p subshell. So this 2p subshell will be filled next. P can hold 6 electrons. We add them into our diagram. There it goes. We see that we have successfully filled up the second shell. Great. Nothing new so far. We move on to the third electron shell, and again, we're going to start with this 3s. So we're going to add in our 2 electrons. You're just really lagging today. There it goes. So we're going to add in our 2 electrons there, right? Following the path, right, it's going to tell us that this 3p is next. So that can hold 6. So we're going to add in 6 electrons. Again, nothing new so far. A second to think. There it goes. All right. But now we start to see difference in how we're going to add electrons in that this 3p, the next one is actually 4s, which means we have to go to a 4th electron shell and add in 2 more electrons in that subshell. Now we only have 1 more remaining, so we're only going to be able to add 1 to that 4th electron shell. Right? If we were to continue this for these larger electrons, which I don't like having you draw larger electrons because A, it gets complicated, and B, that's just a lot of little dots that you have to draw. We would see that from 4s, we would go back to the third shell, finish up that third shell, right? So it's actually at 18. Then we would go to 4p, and then the 5s and the 5 electron shell, and kind of zigzag back and forth, right? So they're really starting to fill unevenly once we reach that third electron shell. Again, I don't expect you to know this for an exam, right? If it's something where it's filling a little bit unevenly, I would give you the diagram, or I would ask you to draw an electron that is simple and is exactly going to follow the way that we have learned. All right, so like a smaller electron, so we're not going out into that fourth electron shell. Any questions about that? I wanted to clarify that because I know that was something I completely forgot about. I haven't talked about subshells since my freshman chemistry class here at Gustavus, so that was very much deeply hidden in my mind. All right, so we learned about carbon in that video. Right, I want you to work with those around you and answer these couple questions about carbon before we continue on. You can talk with one another. All right, so carbon is this amazing atom, right? How many bonds can it form? I see by show of hands, 4, yes. Why can it form 4 bonds? What about it makes it able to form 4 covalent bonds? It has 4 valence electrons, yes. So to fill out that outer shell, it needs to make 4 bonds or share 4 more electrons. Is the configuration of bonds always going to be the same for carbon? Are we always just going to have single bonds? No, we can have double bonds, we can have triple bonds. Right? Carbon can form many shapes, which you saw in that video. Right? So carbon, it's a great atom, right? Because it is, man, it's really lagging. There you go. It is small, right? So it only has an atomic number of 6. So it's relatively small. It's not extremely electronegative, right? So it can be more electronegative than other molecules, but in the scale of things, it's kind of in the middle. Right? It wants electrons, but it doesn't need electrons. It has 4 of those valence electrons, so it can form 4 bonds, like we said. And it can form bonds with a lot of other atoms. Right? It can form a bond with carbon, hydrogen, we see those really commonly. But really any other atom out there that has an available valence electron to form a bond. Carbon tends to be the backbone for many molecules, right? So often we might have carbon bonded with hydrogen, and then other atoms added on to form all of those different molecules that we might see in biology. Right, so super useful. A lot of it has to deal with it just being a small atom. It can fit into many different places. right? And it can form 4 different bonds. It actually gives me a good chance to pause between slides here. I know I talk fast. I really try to slow down, but listen to those videos if you need to go back and relearn something or have it explained to you. Carbon, right? It can form polar or nonpolar bonds. Right? This is also why it's extremely useful. If it's only forming nonpolar bonds, right, and those nonpolar bonds are with carbon to carbon or carbon to hydrogen, right, we would say that these are hydrocarbons. Right? So hydrocarbons are nonpolar. They have equal sharing of their electrons. They're nonpolar. You know that they're going to be hydrophobic, right? And they're not going to be easily dissolved in an aqueous solution. Right? We have an example of a hydrocarbon up top here. You see it's only carbon and hydrogen atoms. A lot of hydrocarbons tend to be like different types of fuels that you might use. Like butane is one that comes to my mind, right? Lots of different fuels tend to be hydrocarbons. Right? But carbon can also form bonds with other molecules, as I mentioned. And those other molecules, depending on their electronegativity, can form those polar bonds. Right? So it has the ability to be polar or nonpolar with who it's bonding to. If it's polar, we know it's going to make it more soluble, right? Water-loving, hydrophilic. Oftentimes what we will see is a molecule having both of these regions. So in this prokaryotic acid here, we see part of it is completely neutral. Right? It's only carbon to carbon and carbon to hydrogen bonds, which are nonpolar. While the other half, right, has some oxygens. They have higher electronegativities. That's going to make this part of the molecule polar. Carbon is great because it can make a lot of arrangements. That video kind of showed you a whole complexity of arrangements that carbon can make. Basically, a different arrangement is going to mean a different molecule with a different function. And so I just had a list of different arrangements that carbon can make. Right? We can have smaller carbon skeletons. We can have carbon skeletons that branch out. Maybe we have a double bond somewhere in here that can change the overall shape and function. And they can form these rings. We can have molecules, right, that we would call isomers that actually have the same chemical formula. Right? So for example, butane and isobutane, both are types of fuels here. Right? If we were to count up the number of carbons and hydrogens, they would be the same. We would say it's C4H10. Right? But their arrangement is different. And because their arrangement is different, they're going to have different properties. Right? So we would call these molecules isomers. Same amount of atoms, right? Same type of atoms, right? But different arrangement. Carbon can also be a part of a lot of different functional groups. So functional groups are groups of molecules. Typically they're branching off of another molecule, but they have the same chemical features and thus have the same properties associated with them. So if we look at an example here, this is an amino functional group. Now this is an example that does not have carbon. Right? But it's a great example of what a functional group is. Right? An amino group is going to be a nitrogen attached to 2 hydrogens. Right? And in this formula, we also see it's attached to this R. Right? That's not an atom. What the R is representing is that this is the rest of the molecule here. Right? But this is the specific functional group we're interested in. This might be like a hydrocarbon backbone, or might be other molecule there, right? But we're only interested in this bit, so thus we abbreviate the rest of the molecule with are. Right? An amino group, right, they're found in amino acids, and they're always going to have the same property, right, no matter what molecule they're bound to. This grouping here is always going to be weakly basic, right? It's always going to be polar, and it can be part of peptide bonds, which is a type of covalent bond between amino acids. We'll talk about that on Friday, right? But the function is always going to be the same. Carbon is a part of a lot of different functional groups, again, making it this super useful molecule, right? So if we just look at a few examples here, I don't expect you to memorize these examples, but if we were to look at a ketone, right, we see it's a carbon double bonded to an oxygen. Right? They're going to be a part of things like waxes and proteins and steroids. Right? Their properties are always going to be the same in that it'll be polar, right, and forms hydrogen bonds relatively easily. Same if we look at a methyl group here. We have carbon attached to 3 hydrogens. Right? Important in molecules like DNA and proteins and carbohydrates, which is the vast majority of molecules. And they're important because they're nonpolar, right? Which then can affect the function of the overall molecule itself. So carbon has a lot of ways that it's useful. Are there any questions about carbon? All right, we're going to do a thought question here, something that I haven't talked about in class, but I want you to give it some thought based on what you know about carbon. So oftentimes, scientists have a hard time conceptualizing other life forms that are not carbon-based. Why might this be the case? Why do they have such a hard time with that? And then as a little bit of a review, these down here are different hydrocarbons, right? I want to know which of them are isomers to each other. So give it about 4 minutes to kind of engage in some discussion here. Maybe our talk will gear towards like aliens and extraterrestrial life forms. How's it going? Yeah. Glasses. Yeah. Ooh. No. We like bring like all of ours. Yeah, we can back Oh, that's classes. You. Yeah, I'm all done. All right, I get the sense that you've already finished and answered these questions here. So, why might scientists have such a hard time thinking of other life forms could exist in the cosmos that are not carbon based? What are some more reasons? Yeah. Yeah, yeah. You have to think outside the box. And, carbon is kind of pretty much everything in our life form. We are carbon-based life forms, right? So it's hard to conceptualize something that we really just don't know about. Right? What are some other reasons? I'm considering this question to read, why is carbon important and so fundamental to life? Thinking about those documents. I'm going to call on groups then. I'm going to call them as a group in the corner. I know we discussed, so I know we have an answer. Yeah, carbon is a super successful atom in terms of the amount of connections it can be made, right? It's small, it can connect with everything. It forms a backbone to so many molecules. Either we don't know some properties of molecules out there on other planets, right? Or there might be something that exists that we haven't discovered yet. Who knows? But we just have a hard time conceptualizing it because on Earth, carbon is just so predominant. All right, and then a little bit of review on the bottom here. Which one is an isomer with this first one? The one on the right, the fourth one. Yes, I heard some murmurs in the crowd. Right, so these first two are isomers. Are the middle pair isomers? No, they're also isomers, right? If we were to count out the number of carbons and hydrogens, the first and fourth would be the same, but they're different arrangements. Same with these middle pair. All right, we're going to have a lot of group review in class today because it's just a great way to live, right? But we're going to start moving forward and talking about biological molecules. So we talked about carbon, and we're going to see carbon in all of our biological molecules. Many of our molecules that we will look at, right, tend to be made up of building blocks. Right? So we might have monomers. So monomers are a small molecule. Right? This is like the basic building block. Think of it as like one Lego piece. Right? We can stack many monomers together, right? Like we're building something in Lego here, to create polymers. Right? So polymers are built by stringing together these smaller monomers. Okay, so my very abstract example up top here, we have our monomer, and we can keep adding to it one by one. These transitions are not going to be so slick anymore because it's lagging. one by one to build this long chain of polymers. Right? Now, if this polymer is huge, right, we're thinking like thousands of monomers long, right, then we would call it a macromolecule. Right? It's just a very, very large polymer. So to put this into like a real world example here, we might have a monomer, which would be glucose, making its appearance. There we go. We might have glucose. Right? We can put many of these glucose molecules together, right, string them together to create a polymer, which in this example here would be a long chain that might be something like starch or different fibers. Right? We're going to see this for a lot of our biological molecules that we will talk about. Right? But how are these monomers actually working together with one another, right? Well, molecules are going to be bound to one another. It's going to involve some type of chemical bond, right? And because they're bonding together, it's going to involve a chemical reaction. So chemical reactions, specifically ones that we call the polymerization, right, is the process of bonding these monomers together. Again, we see separate smaller molecules. They are going to be bound to one another. Right? In this case, they're all covalent bonds. And we're just going to keep adding and adding and adding in this process of polymerization. Right? So that's the technical term for how this happens. But the actual chemical reaction itself, right, has another term, and we would call it a dehydration reaction. Right? If you think of the word dehydration, probably think of something with water, right? Hydration, water. Dehydration means you don't have enough of water. In this case, a dehydration reaction is a reaction that takes away a molecule of water from two monomers and leaves behind a bond in its place. So if we look at our monomers up here, right, on either end of our monomers, we have a hydroxide group and a hydrogen. Now, if we were to link them together, we could take the hydrogen side of one and the hydroxide side of the other. Right, together, they would form water. And when the water molecule is formed, right, it leaves behind a bond and an excess water molecule that will then go away from the reaction and do something else in the cell. Right, so a water molecule is removed. Right, and this process is going to repeat. We'll see how this actually plays out in my little simulation here. All right, just as kind of like an animation here, we have our monomers. Right? We have hydroxide on one side, hydrogen on the other side. We're going to combine them together. That's going to release a water molecule. It's kind of laggy. Still kind of working though. Releasing a water molecule, right, and leaving behind a bond in its process. Then this will be catalyzed by enzymes, which are proteins. We'll talk about those on Friday here. So we've built the monomer, or we built the polymer, built a polymer out of monomers. But how do we break apart the polymer? Right? That is done by exactly the opposite reaction. Right? So dehydration reaction, we're taking away a water molecule. In a hydrolysis reaction, you're adding in a water molecule. So now we're starting with a long chain of monomers. We're starting with a polymer. Okay? That polymer is going to be broken apart, right, by adding in a water molecule. Right? That's going to add back in a hydroxide group and a hydrogen group and slowly break apart the monomers from one another. Okay? Again, this process repeats over and over and over. And is catalyzed again by enzymes that make this process go a little bit faster than it would naturally. So hydrolysis here, we have a polymer. We are going to introduce a molecule of water here to this covalent bond, which will then donate hydrogen and a hydroxide group. Right? Once it donates that hydrogen and a hydroxide group, the monomers will split and that bond will break. Right, so exactly the opposite of how monomers or how polymers are built. All right, do a quick group review here. I want you to look at all these reactions. The bottom one is a chemical reaction that's not listed out, right? But you should still be able to pick up on the clues too. Is this a dehydration reaction or a hydrolysis reaction? I'll give you a couple of minutes. What reaction is that?
Hydrolysis you're adding. Oh yeah.
Yeah. There you go. All right, our first reaction here. What type of reaction is it? Dehydration. Yeah, it's a dehydration reaction. And we know it's dehydration because not only are we combining two smaller monomers together, but we're also releasing water, right? We're kind of dehydrating those monomers, right? What about reaction number 2? Hydrolysis. Yes, I hear the whispers. Hydrolysis. Again, we're taking a larger polymer and breaking it down to its component parts by adding in water. What about this last one? We're digesting proteins, carbohydrates, or a lipid. What type of reaction would that be? Yeah, it would be hydrolysis because we're breaking it apart. Oh, I have the wrong thing there. It is hydrolysis because it is breaking it apart. All right, so ignore that there. I forgot to change it. Right, the key here is we're taking the larger molecule, like proteins, carbohydrates, right, and we're digesting them and we're breaking them down. All right, we're going to dive into some of these major types of organic molecules. We're going to start with 2 of them today. All right, so we're going to learn about carbohydrates and about lipids. All right. I like this table. I put it up here because it's a good reference to go back to. Right, but we're going to elaborate on what's on this table. So we're actually going to start with carbohydrates. Carbohydrates they're typically going to be composed of mainly carbon, hydrogen, and oxygen. They do follow that monomer to polymer kind of rule here. The monomers, in this case, we would specifically call them monosaccharides. Right? Monosaccharides are sugars. Right? So sugars are the monomers for our carbohydrates. You can write out sugars in a formula where they're just in a line here. Right? This is less commonly found in many different organisms. More commonly, we will see sugars in this ringed structure. Right? So monomers, we're talking about like galactose or glucose or fructose. Very similar to each other, right? But there's just slight differences that make them slightly different molecules. But they're all sugars. Right? Monosaccharides, really great in powering your cells. Right? They do a good job of giving your cells energy so that the cells can perform whatever function that they need to. Right? But they're also a monomer, which is a very small molecule that can be combined together to form other larger polymers. So these monosaccharides can be used to manufacture other molecules that our bodies might use. One of which way that they do this is by creating disaccharides. So disaccharide is two monomers joined together. Right? In this case, it's two monosaccharides, so two sugars. They're going to be bound to each other, right? Just like we've learned. We're creating a polymer, making it longer, so it's going to be a dehydration reaction, a process of removing oxygen to create that bond. And the specific bond formed in this case, it is a covalent bond, but we would call it a glycosidic bond. And the reason we call it something different isn't because it's not a covalent bond, because it is, but it's specifically a covalent bond that joins a sugar molecule to another molecule. Right? So this could be a sugar molecule to a sugar molecule, or it could be a sugar molecule to something that's not a sugar. Right? But if it's involving a sugar molecule and it's a covalent bond, we would say it's a glycosidic bond. All right, so we have our monosaccharides, we have our disaccharides. We also have polysaccharides. There are many different types of polysaccharides, and these molecules are huge. They are a macromolecule. They consist of thousands of monomers, thousands of sugars linked together. We might get such polysaccharides like starches, glycogen, cellulose. They all have very specific functions to them. And these functions are ones that I'm going to have you do a little research on and figure out what do they do. So you might have to use your computer to try to figure out what is starch and why is it biologically important. Right? How do organisms use starch? Same for glycogen, cellulose, and chitin. What do you think the word starch? Polysaccharides? All right, so we have 4 different macromolecules here, 4 different very large polymers. Why is starch important? What does it do? Yes, stores energy for plants. So a lot of plants, like you see starches in potatoes or cornstarch, right? Stored in those plants, we will utilize that stored energy when we eat those starches. What about glycogen? Yeah, another type of energy storage. This time in animals and in us as well, right? So it's a way for us to store energy until we actually need it. There it goes. Right, so energy storage as well. Cellulose. What role does cellulose play? This is moving into my realm of expertise. Yeah, plays a huge role in structure for plant cell walls. Cellulose is often very hard for animals to digest, particularly us. Though some animals have adaptations to their digestive system to be able to more easily break down cellulose. But it provides structure for our plant cells. And then lastly, chitin. What does chitin do? It's something that we eat. It provides some sort of purpose. Anyone on this side of the room want to answer? All right, I'm going to call on you because I know we discussed it.
It provides like protection layer for insects.
Yeah. So it's part of like the exoskeletons of a lot of insects, right? It's that layer of protection that we might see. Right, there's my little picture here. We can see this insect has molted, right? This is part of the exoskeleton that's left behind. It can also be found in the cell walls of fungal cells too, again providing structure. All right, we are going to move on to another group review. This one a little bit more involved in that I want you to put your carbohydrate knowledge into practice. So here I have the ingredients for 3 different food items here. I want you to find the carbohydrates and then fill out the table on this next slide here. So again, you might have to use the internet here. So on the next slide, it's asking you, right, based on the food item, it might pop up here, right? How many carbohydrates are in the ingredient list? Are any of them monosaccharides? Which ones? Are any of them disaccharides? Which ones? And are any of them polysaccharides? Which ones? So I imagine this will take a little bit. right? It's a good practice here to try to look up these chemical structures. So I'm going to give you a good 10 minutes, and we'll see where we are. So has everybody had a chance to kind of finish up in those 10 minutes? Was that enough time? Maybe some of you have, some of you haven't quite. But I think what we'll do is come back together as a group and review it. So, if we were to fill out this table here, soft drink, how many carbohydrates are in the ingredient list of soft drinks? I hear 2. I have a 1. What was the second one that we had saw? So I had to do this exercise so I'd be able to give you the answer. So I don't remember all the answers off the top of my head. I saw the 1, which is the high fructose corn syrup. Is high fructose corn syrup a monosaccharide, a disaccharide, or a polysaccharide? Shout out the answer if you know, or hold up your fingers. It looks like it could be a disaccharide, but it's actually a monosaccharide, right? It's made up of 2 different monosaccharides, glucose and fructose, that are not joined together. Right? So high fructose corn syrup are monosaccharides. Right? Now because we only have 1 in our soft drink here, that means we don't have any disaccharides, we don't have any polysaccharides. What about those wafers? So I specifically looked up like an ice cream sandwich and gave me the ingredients for ice cream, gave me the ingredients for wafers. I chose just to focus on the wafers here. How many carbohydrates were in that ingredient list? Let's see, 4 by show of hands. Let's see what I got. 1, 2, 3. It's 4. Yeah, I put the wrong number there. So 4, right? So we have our bleached white flour, we have sugar, we have dextrose, and we have corn flour, right? Which of those are monosaccharides? Are any of them? Which one? whispers, but I can't determine what you're saying. Is it the flours, the sugar, or the dextrose? Yeah, the dextrose here. So dextrose is a monosaccharide, so when you looked up its chemical structure, right, you only saw that's one sugar, right? It's not connected to anything. The disaccharides here, right, I'll kind of skip ahead here. We have one, it's sucrose, right? Sucrose is the combination of glucose and fructose that are combined together into a disaccharide. And the polysaccharides are obviously the ones I haven't listed yet, right? These would be the flours, right? These flours, they're starches, right? Starches are polysaccharides built up of many thousands of sugar molecules. All right, this honey almond granola. How many carbohydrates did we get in this ingredient list? Hold up your fingers with the number that we counted. All right, I heard 6. I saw 6. All right, I got 7, and let me make sure I counted this one right. 1, 2, 3, 4, 5, 6, 7. So I have 7. So maybe you might have included this barley malt extract. Maybe you didn't. Sounds kind of an oddball there. So are there any monosaccharides? A very reserved group today. There's actually 2. So molasses and honey, they're both made up of glucose and fructose. Right? Not combined together, just separately, kind of hanging out. Disaccharides. Which one of these might be a disaccharide? Somebody from the middle row, if you found that answer. Do you have a guess? Cane sugar? See what my thing says. Yeah, cane sugar is one of them. I also listed molasses. I don't remember why I listed molasses, but I wonder if molasses is also has a disaccharide with the glucose and the fructose. And that barley malt extract, oftentimes that's maltose, which is a disaccharide as well. Right, so still all sugars, right, which leaves the remaining 3 here, which would be our oats, our flours, right, consisting of starches and different fibers. Right? It's kind of that have your daily fiber in your breakfast kind of part of these carbohydrates. Yeah. Yes, that caramel color would maybe count as the, yeah, I did not put a color in there because I'm like, why bother with color? So that was my oversight. So yes, awesome. It's done. Our class is done. So have a good rest of class. If you look, there is a homework. It's just a 2-minute video. If you get to it, great. Otherwise, we didn't even finish this lecture, so I'm not worried if you look at it. But keep in mind that questionnaire is due on Friday, and my last office hours are today. Yes, yeah. Yeah, you too.